So our next speaker is Michael-Christopher Keogh. Um, he originates from Ireland, where he did his bachelor's in microbiology at the National University of Ireland in Galway. He then moved on to London, where he did his PhD in immunology at Imperial College London, and then moved to Harvard Medical School, where he underwent his postdoctoral training with Steve Bertowski, where he was a part of just an amazing amount of really cool studies working on understanding the writers and the readers of various histone post-translational modifications. After his postdoc, he moved to Albert Einstein College of Medicine in New York, where he was a principal investigator, and then subsequently moved on to Epicipher, where he is now the chief scientific officer. And independent of any sponsorship that Epicipher is providing, it's been extremely impressive to see the work that they have done and their commitment to continue publishing their research so that it's available and public for all of us. And I think that's evidenced as well by the fact that he volunteered to come give a talk with us here. So Michael, thanks for being here, and please take it away.
Thanks, Jackson. And also, thanks everybody for coming out. I think I, in fact, even saw Steve in the audience, Steve Bertowski, my old mentor. So as Jackson mentioned, sort of Epicipher has continued sort of doing a large amount of sort of foundational academic research because we've been supported in our R&D teams by the NIH. And what I'm going to show you today is sort of the work that in our original programs, our first love was trying to sort of fix CHIP all those years back before we moved to cut and run, trying to basically identify reagents that were good and bad, where the long-term strategy was always to try and sort of map the PTM landscape. And in this case, what I'm going to show you then is the beginnings of that original PTM antibody characterization platform, the fact that how much we learned when we were going through it. And then once we had good antibodies, the ability with the collaboration with the Immunological Genome Consortium to actually map the PTM landscape in the primary immune system of mouse. So the take-home message, I mean, I always like to put this first in case people tune out. They might pay attention to my talk for at least a minute. And so is that what we've shown is some very bad news, which is some about 70% of histone PTM antibodies failed testing. In the middle of this, there was also some very good news because we feel that we understand why this had happened. And we also identified a path to actually make it such that the next generation of antibodies don't have to fall into the same trap. And I'm going to show you some positive data in that regard. And then the idea is, okay, once you've got these good antibodies, what you can apply them to novel biology, or you can apply them to exploring at lower input applications. And then in the latter case, it was trying to do automated genomics to trying to map the immune system of mouse. And again, as I mentioned, through the collaboration, an extremely good collaboration with the Immunological Genome Consortium. So this audience does not need me to tell you what the nucleosome is. I like showing it at the beginning because I think it tries to explain to people why what happened perhaps. The standard way that we look at it is, the DNA 147 wrapped around the nucleosome. The tails are shown extended, and historically, for a lot of reasons, this is done. I actually like showing this alternate viewpoint, which is simply looking at the charge of the actual what's comprising this complex entity. And what you immediately see is the DNA is negatively charged. You also have this negatively charged acidic patch. But what you see is the tails are not. The tails are positively charged. And what do positive and negative charges do? They attract each other. So in other words, this entity, which is depicted as sort of splayed open like a spaghetti monster, is not. It's wrapped back upon itself. And this is profoundly impactful on biology. And I think when we keep this in mind in everything we do, it sort of helps us to explain what we're actually looking at. And over the last few years, we've been, the other thing I would call attention to here is that you can, in fact, look at the tails and understand where the positive and negative, where the positive charges are coming from. And they're largely contained inside the arginines and lysines. You can neutralize the charge on the lysine by acetylating it. And there's, in fact, entire branches of biology, the first modification that was identified. In one part of my talk, I'm going to talk about a modification that neutralizes the charge on the arginine, and that's citrullonation, which was definitely flavor du jour for about a handful of years when the field was very interested in arginine methylation. and then sort of fell out of favor. Part of the reason being because it was extremely difficult to map the modifications. But I'll just show you, this is one of the new pieces of biology. I'll do 2 quick slides on that. So over the years, we've worked on extensively with colleagues on looking at the impact of neutralizing tail charge. And others have demonstrated that nothing engages the nucleosome on its own. Everything is combinatorial engagement. The histone code sort of focuses our mind on things like, oh, 2 PTMs together or 2 PTMs in opposition to each other. But you can get combinatorial engagement in multiple different ways. And so these surfaces are profoundly impactful. They attract or repel things. And so the histone PTMs in primary sequence, or you can, of course, have the wrapping and linker DNA. That's a great opportunity for binding potential. And you can have the nucleosome acidic patch. This impacts all the readers that we like interrogating so much. And it's absolutely no great surprise to discover that the antibodies that we're trying to use in genomics assays and in other types of approaches are, of course, directly impacted by this as well. All right, so simple sort of transit point. What we want to do in this what we're trying to drive through in this talk is the map of the immune system. But to do that, you want to make sure that the antibodies you're working with are actually good, because if you say K4-trimethyl is K4-trimethyl, then you have to trust that that statement is correct. And so over the years, Epicipher has been very generously funded by the NIH, and we acknowledge that completely that they have been transformational to the way that we've grown Epicipher and the things that we've been allowed to do. But they've allowed us to build nucleosomes, and being able to build nucleosomes therefore allows you to interrogate chromatin. It gives you the basis of binding assays. It gives you the basis of genomics, allows you to standardize genomics assays. And so the 2 things I will call attention to are our ability to make nucleosomes with post-translational modifications. These are not analogs. They're not enzymatic products. They're the actual modifications themselves. They're made semi-synthetically. Like Tom Muir, for example, has been profound leadership in this field, and Tom and others have demonstrated that this is possible to do. And essentially all we've done is done it on a much larger scale. The other thing we can do is the wrapping DNA. We can modify it by just knocking in barcodes. So when you can do these 2 things together, you can make PTM-defined nucleosomes, which can be identified in a pool in various ways. The simplest way being in a genomics assay is to simply look at the wrapping sequence. So once we've learned how to make these various forms of nucleosomes, for the antibody characterization, it's not simply enough to say, okay, I have an antibody to K4-tri. I look at K4-tri and the unmodified forms. We said, no, we're going to do it in the form of panels. And so here the lysine methyl status panel is K4, K9, K27, 36, and H4K20 as the unmodified mono, di, and triforms. In other words, a broad swathe of biology and that we're now coming to appreciate. Originally, I think it was fair to say we were very focused on a certain number of modifications like K4-tri or K9-tri or K36-tri, but we now know that there's many others and their biology is very important. By the way, everything I talk about, every time we've done these characterizations to a PTM class, we've always done the same approach. That I think in the case of the acyl stat panel, for example, that had 25 different nucleosomes representing various acyl states, either alone or in combination. So we screened antibodies to this, and I've been talking about this down the years, and essentially the results were bad. About 70% of antibodies fail testing to these standards in CHIP. And by fail, I mean that they either show unacceptable cross-reactivity or unacceptable recovery. So this is easy to understand. I'm to K4-tri, I see K4-di, 25%, I fail. Recovery is something that we're particularly interested in, because the recovery is if I give the antibody 100 molecules, how many will it actually recover for me? That matters because long-term, if we're interested in low-input applications or single-cell work, you cannot do that with an antibody that's inefficient. And so we've continuously monitored both metrics down the years. This, by the way, is not based on 5 to 10 antibodies. This is based on 400 on this original data set. We now have data on approximately 1,000. We have data on the original, on multiple lots. We can see lot variability. We can see, programs change in terms of their behavior. It is profoundly problematic across the board. The other thing is that I will call attention to the fact is the highly cited antibodies, the failure rate there is about 80%, and that's the top 30 highly cited antibodies. And the reason that they mostly fail, I think, is they have been selected for efficiency scores. What people are looking for is continuous yield. Continuous yield, it means experiments that I can actually analyze. But sadly, you can drive yield forward by having cross-reactivity profiles. Because now instead of going after one target, you're going after more than one. It wouldn't matter so much in terms of the data if it was purely on the standards, but you can actually see it in the tracks as well. So in other words, the standards are a demonstration that this is a profound problem. And this is from a paper that we did some years ago with Alex Ruthenberg and Scott Rothbard. And I'll just call attention to these antibodies to K4 tri. The red bar says that's set to 100. That's the target of these K4 trimethyl antibodies. But what you see for these two here is that they are cross-reacting. The blue bar is quite high. That's K4 di. That cross-reactivity level is 60 to 80%. That is a profound problem if you're mapping K4 tri in these experiments, because you're looking at tri and di. Also, in this case, the standards are equimolar, but inside cells, the PTMs themselves are not. So PTM mass spec data has allowed us to understand the relative abundance of each of the forms. Tri is a very rare mark. We love it a lot. It's all piled into the same place, these tight, high amplitude peaks, but it is a very rare mark. Di in comparison is much more abundant. And so therefore, an antibody that does this is looking at more than just tri. It's looking at tri and di at the same time. And you can see this in the track. So this is the K4 di track in blue. These two antibodies here are ostensibly K4 tri, but that's these ones. And essentially, when you do have an antibody that is specific for K4 tri, what you now see is the disappearance of peaks. That, by the way, is also a disappearance of yield, which again is this idea potentially during early antibody development, when the first antibody arrives to a given target, we have a feel for how much I should yield. We have a feel for the peak structure I should get. And now if a new lot appears that loses most of the peaks, okay, which one was right? And it's just always something to keep in mind. Okay, so how did this happen? It's probably because of the peptides. Every antibody to a PTM is raised to a peptide. It's then continuously screened to a peptide. Genomics is the last thing it sees. If it's actually screened in genomics, that's an impressive feat. But what we showed with Alex and Scott was that there was minimal predictive correlation or predictive value between the peptide data and the nucleosome data. Peptides are not nucleosomes. Because remember, the antibodies are now going to be challenged in a completely different environment. And the question is, what will that do to their capability? However, what we searched for years and we said, well, okay, we probably can make a version that is more nucleosome-like. And this is a Luminex-based approach. Essentially, we have these color-coded beads onto which we can immobilize nucleosomes. So now suddenly I am presenting the antibody with the right target. The other thing is I can multiplex these things, so I can do a competitive landscape, and I can wash them as if it was a chip or a genomics experiment. So by doing this approach, which we internally call Captify Nuke or Luminex Captify, you have this nucleosome compatible platform which you can multiplex, wash, high throughput, and high sensitivity. Now the question is, can it actually predict with high correlation what the genomics would do? That is Captify Luminex data to a collection of 25 antibodies. So what we're looking at, each antibody is a row and the columns are the various targets. And you can see, it's a heat map. You can actually see the binding profiles of each of these. That is the SNAP chip data, essentially the spike in chips. And what you can see is a very similar metric. It's not identical, but it's very, very similar. The other thing is that we looked at, well, why is it not identical? I mean, how close can we go? So we looked at the efficiency scores and we were binning them by whether the antibody was more than 10% efficient or less than 5 sort of thing. And what we discovered was that the highest efficiency antibodies had almost perfect predictive value for the Luminex-based approach. As the antibody became less efficient, then there was a disparity in the data. Now that comes back to, remember, we're only looking for high efficiency antibodies anyway, because the high efficiency antibodies are the path to actually the low input applications or the single cell work. So this didn't really cause us any major problems. But it is, I will call attention to one specific thing. This experiment took an afternoon. This experiment took months because we were screening through, at the time, we could do it much faster now, but it's also a much more expensive approach. So this was looking back at what had come before. And in fact, we've now released all of this data. It's all available to you now, but it's a snapshot of what the state of affairs was approximately 4 years ago, because this is data, we've been sitting on this data for years. But we have released all of it. It's accessible. Keep in mind that don't simply pick the best performing one then. Every data, a piece of data is lot specific, and we've seen changes in lot behavior. But going forward, what we've also done is we've moved to other types of genomic approaches. And so, for example, we discovered cut and run. And in fact, that's one of the things we've run with over the last few years is this new technology. We immediately made the standards compatible with cut and run. We immediately got them into the experiment such that we're now monitoring the spike-ins and we're monitoring the performance of the cut and run experiment. So in other words, before we did any cut and run experiments way back when, we immediately went back to our freezer and said, okay, we had all these chip antibodies. The question is, which of them actually work in cut and run as well? And so that gave us a very rapid reappraisal of the landscape. And here, for example, is a collection of high specificity antibodies within K-Metstat. Again, what you can see is on target is in blue and off target. So we should reverse this heat map. But the idea is that you can actually see, as we found in this panel of 15 targets, we found 14 of them capable antibodies pretty much straight out of the freezer. It was a lot of work. And the other thing is, we knew going forward is we were terrified about the fact that we were going to lose these antibodies because they were specific lots. So over the last few years, what we're now doing is applying everything we learned from the Luminex-based approach and using that to actually select new candidates. They get to see the nucleosomes through the process. We're not only interested in specificity, we're also interested in efficiency again. And in the case of a cut and run experiment, the simplest way to do efficiency testing is to do it at your standard 500,000 cell input and then drop the input by 10x and ask, do I still recover the same peaks? If you don't, that means that antibody is not going to be suitable for low input applications. Here we see two antibodies, K4 mono at 500,000 and 50,000, essentially at the same peak structure. Here I lose most of them on this target to K9 dimethyl, or sorry, K9 acetyl. So what can you do with these new sort of best-in-class antibodies? You can do two things. You can explore new biology. You can potentially find targets that had failed. In other words, every antibody that was out there didn't work. And so now you can make a new antibody, hopefully, with this new approach and go after that biology. Or you can apply it to lower input applications. I'm going to show you examples of two. So the first one, coming back to citrullination. I mentioned it in passing. A very hot topic for a couple of years in the PTM epigenetics field. It's now talked about a lot in the mitosis field, but in the PTM field, chromatin biology was very flavorful for a while. One of the interesting things about it is it neutralizes charge. And so that's got massive impacts. If you citrullate arginine, you open up the chromatin structure just like the same as you do with acetylation. We screened every antibody to citrullination. We couldn't find one that worked in genomics. And that was not to say the target was there, but we certainly couldn't find one that actually behaved particularly well. I think there was one target, but it didn't give us genomics data. But we said we had access to NIH support, and we went in and we created more. And essentially, we went in with immunized with a peptide, but then in collaboration with Bethel Fortis, we were doing recombinant nucleosome. So we went and looked at the recombinant clones, and we challenged them early and often to nucleosome targets in our Luminex-based approach. And we didn't just challenge them to one of the targets we were interested in, because on this tail, 3 of the arginines are citrullinated at least, and that's R2, R8, and R17. And so we basically created a panel of nucleosomes that contained each of these, R2, R8, and R17, along with a hypermodified. And out of the various clones that we got some exceptional clones. This is just some examples. They absolutely adore R2, ignore R8, R17, ignore the unmodified. They do like the hyperacetylated form, by the way, but most antibodies, when they're challenged with that, actually do prefer the hypermodified forms. But we found good targets for each, and then we went into genomics. And I'll just give you a very quick 2 statements. R2 didn't give us any tracks, but R8 and R17 did. And in other words, here is the R8 and R17 track in a relatively large window. It didn't have any relationship to what we were usually used to looking at, which was active genes, K4 tri, K36 tri, or polychrome regions, K27 tri. Well, what was immediately apparent in the data was that it appeared that wherever K9 tri was, R17 and R8 sit weren't. And that was a very interesting sort of inflection between the two PTMs. And in fact, you can look at single windows, but that's dangerous. So here what we've done is we've binned all the data by K9 tri-peaks. And what you see is in these windows, therefore R17 sit is dramatically absent from that location. And vice versa, if you enrich for the R17 peaks, K9 tri is absent. And in fact, you can look at the regions where the two would join together. And you see where the K9 tri peak ends. Basically, there's a plummet. It doesn't bleed into each other. Essentially, the two things seem to stay far, far away from each other. This is entirely new biology. I, by the way, do not have an answer for why this is. And we can, essentially, if anybody in the room does, I mean, we're planning on releasing this data in a few weeks. Okay, so for the last sort of 7 minutes of the talk, then I'll go to the other opportunity. And that is, I now have these amazing antibodies. The question is, can I use them to actually map the mouse immune system? ImGem came to us some years ago and said that this consortium has been mapping the immune system at various levels at various times based on the technology that was existing. So they originally were doing it with microarrays, and now they were trying to do it with essentially a cut and run or a chip-based approach. The one thing they threw us with for a sort of a curve was they said, by the way, there's going to be about 90 different immune cell types, and you will only get about 10,000 cells per because they're multicolor sorts. And this is very precious material, and you're not going to be able to give it an opportunity to make mistakes. And so essentially what we were being asked to do was around 2,000 cut and run reactions, where we were working with 2% of the standard input. And therefore, we wanted to be certain at any given point that the data that we were looking at was actually due to biological distinctiveness, as opposed to a technical problem on a given experiment on a given day. And so for that reason, through this process, we automated cut and run. And that's actually what's actually used in our lab right now. And so we looked at the entire pipeline and said, gee, this is the way it's organized. There were various ImGen labs, all of them specializing in various cell types, in various cell types, and they were doing FACS, isolating the material, and then lightly fixing it, FACS analysis, and dropping it into a nuclei isolation cryopreservation buffer, shipping it to the Broad, the central lab where the material would be logged. It would then be shipped to us, and then we did what we termed the dilute and dispense protocol. We simply didn't count anything. We didn't look inside any of the vials. We dropped them into 96-well plates, added the antibodies and the reactants, and then just hit go on the robot. We took it from one robot to another. We did the original cut and run on robot A, and then basically moved it to robot B for the library prep, and then shipped it back to the Broad for the sequencing and data analysis. So this program ran over about 20 plates over about 2 years. And we knew that it was going to be this scale going in. So we wanted to, like I said, again, you're always terrified that what you're looking at is noise versus real data. Because also you're not, I think every sample, I think, was run through at least twice. But we looked at every parts of the standard cut and run workflow to try and see if there were points where we could automate it. Also to try and see if there were points where we could drive up yield. Because one of the things we were always concerned about was there's a multi-point process. And therefore, if there's any steps in it that are inefficient, they'll undermine the entire thing. So we looked for points of hyper efficiency gain, and we also looked at points where we could investigate what was going on intermediate. We stopped doing that, by the way, after a while. There was so little material, we felt it just wasn't helping us in any way. And so we optimized the buffer components to drive up yield, and we also optimized the MNAs, or protein AG MNAs, to drive up yield as well. And then, of course, just because you've got higher yields, that doesn't necessarily mean that that's due to increased signal. You could have a higher yield simply because you've driven up noise. And so here's just looking at the standard and the optimized conditions for IgG, K4, K27, tri, and acetylene methyl. And basically, we're getting essentially the same peak structures. We have made this thing better. We've managed to eke more material out of it by basically tweaking the system, and we're not doing it by reducing the dynamic range of what's going on. So we then went back to all of the antibodies we'd seen before, and they were all tested at a much higher input application. So we essentially brought them all down to 10,000. And what we ended up with was what we termed the Elite 8. By the way, this was done about 4 years ago or 5 years ago. If we'd done that same experiment now, I think we would have had many more candidates because we've been able to create more potentials because of the nucleosome-based screening. And so what this gave us was K4 mono and K27 acetyl that was associated with enhancers. We got K4 tri for active promoters, K27 tri for polycomb, K36 tri for active gene bodies, H33, which is a mixture of things, and then CTCF, which is the sort of new higher TADS, basically higher order chromatin structure. Each of these was capable at 10,000. One thing that was missing, we would have loved to have had, for example, in this program, K9 tri, because that would have given us repetitive locations, but we couldn't find an antibody that actually passed for that. We looked at the nucleosome standards, and what you can see here is basically got this amazing behavior. That looks great. And then we looked here essentially just to show is each of them gives you exactly what you expect. Fibroblast, this location here is commonly expressed. This location is expressed in fibroblast and polycomb repressed in B cells and vice versa. So essentially you're getting the peak structures you expect. And then to set it up to actually make sure that you can track what's going on over time, what you have are the unknowns on every plate, and you've got a tracker column on every plate. So in other words, every single plate got a known entity, such that if the fibroblasts failed, that was a demonstration that the samples, essentially the plate was to be discarded. So we could actually track everything over time. So over the course of 2 years, we also kept track of the nucleosome spike-ins. We also kept track of the metagene at various locations. And what I can tell you is that over 2 years, we failed one plate. We were also, the drift was minimal, so therefore we were confident at the end of it that the biology on plate 1 and plate 7 and plate 12 are real, and they're not driven by this sort of hanging out at the edges of what the technology is theoretically capable of. And this is the final slide. Here, what we're looking at is just one way of looking at the data. So qualitatively, did we get full coverage of every target in every lineage? And the answer is no. So green are our passing data sets, red are failing data sets. And we look at it in this way because it gives us an opportunity to understand, were any of the antibodies weaker than any of the others, or were any of the sample types refractory to the approach? So what you can see, for example, over here, there's a lot of red in the macrophage monocyte lineage. That's possibly because those things are full of nasty lytic granules and they're fighting against the approach, and or it's because they're difficult to count on a fax because of their morphology. The data normally looks like this, which is utterly tedious to look at. I mean, you can't explore biology like that, but our colleagues at ImGen and Steve Josefowicz's lab and others have been looking at this, have been playing with this data set now for a couple of years. And so we're able to look at, well, okay, how do you interrogate biology? And I'll just show you one example of what they were able to do. Here, what we're looking is termed the lineage specificity index. Basically says across all 90 cell types, looking at peak location, are the peaks in the same place in every lineage, or do they move in one lineage or another? In other words, what is the tissue specificity of each PTM in each cell type? What you see around here for K4 tri or K36 tris, there's this big peak. In other words, genes are in the same place most of the time. They're not moving around. That is the bulk of the material, but the tail is where a lot of the biology is actually lying. That's, of course, the tissue-specific genes. And so those are moving around and they're appearing in one cell type and not appearing in another. That makes absolutely perfect sense. That's what we intuitively understand. The one interesting thing is K27 tri has a bit of a broader domain, but polycomb doesn't appear to be driving the distinctiveness between lineages when you're looking at the PTM landscape. What you actually see is that the distinctiveness across, the greatest degree of difference across all of the lineages is where the enhancers are. And this is the K27 acetyl and K4 monotray data. What this essentially says, and this is sort of the way we've sort of thought about it internally, you've got the same set of genes that are expressed in let's say 5 or 6 different lineages. That will show up in where the K4 tri is and where the K36 tri is and where the K27 tri isn't. But there's multiple ways of switching that gene on and off by using different enhancer combinations, different transcription factors involved. So in other words, the biology is being driven by where the enhancers are and with the transcription factors that are sitting inside those locations. The K4 tri, et cetera, is an indication of basically RNA-seq type data. That's the output, but the input is actually what's driving the system forward. The TF guys will love this because that's what they've been telling us for all these years. I will stop there. I will acknowledge the support of all the collaborators who have alluded to through the talk, but I also, again, the immense support that we've been given by the NIH, the flexibility they've given us to do this sort of biology. Most of what I've shown you hasn't yet been published, but look forward, hopefully, in the coming weeks. I apologize, I ran over by a minute. Thanks, Jackson.
No worries. Thanks so much. I'll start off by asking about the citrullination. It was only 2 slides, but it's obviously fascinating. I mean, the counter enrichment with K9 trimethyl would suggest that it's kept out of like the really condensed chromatin regions, but it did look like there was some co-enrichment with K27 tri.
Yeah.
So do you also see it, like if you look at borders of like the K27 tri domains, or if you look at borders of active domains, do you see similar counter or co-enrichment?
No, it is truly, I mean, that one is a very interesting, I mean, we don't understand it internally, the biology of this. We truly don't. And in fact, that was the first thing we always do is we look at the land. Every novel PTM that arrives in the lab, we compare it to everything that we've seen before. We've got about 60 or 70 tracks now because we've got all these new antibodies. And we saw nothing that was associated. There was no relationship to gene bodies, gene promoters. Polycomb didn't matter. K9 tri was distinct. Now, you could argue that, yes, it's accessibility, and it might be. The one thing we also did was we looked inside enhancers and K4 tri promoters. There's no hint that the higher level of accessibility at those locations is driving citrullination. So in other words, if it's an accessibility issue, it's above a given threshold, and an open enhancer doesn't bring it up any further. So it's like it's either a muscle twitch, it either does or doesn't. But biologically, we have no idea what's going on here. We truly don't. And it's going to be one thing we can say, because the most scary thing was it's simply because the antibodies are blind to combinatorial. In other words, an antibody cannot see K9 tri and R17 CYT. I didn't show you. The panel was much bigger than just the single citrulinations. Nowadays, we normally also look at combinatorial signatures to try and identify antibodies that are independent of as much of the ongoing biology as possible, that they're not biased by oh, if this thing is nearby, it basically ablates the signal, even though that's a different branch of biology. And therefore, we're very careful about that. So it's at least not that.
Right. That plays into, there's a question in Q&A from Mark asking whether the K9 trimethyl would block the binding of the antibody to the
No, and also K9 trimethyl also doesn't impact the activity of one. There's 5 PAD enzymes, the peptidyl arginine deaminases. The one that's studied the most often in the context of nuclear citronylation is PAD4. And we've done a lot of enzymatic assays. PAD4 is highly promiscuous. It's blocked by pre-existing arginine methylation, but it does not care in the slightest about the extant PTM landscape. It doesn't care about transcriptionally active regions in terms of all those PTMs. It doesn't care about any of the other locations. And it absolutely does not care about K9 tri. It happily modified independent. Those are, of course, in vitro assays.
So then, I guess, how abundant is it? Did you do a similar calculation like you did with the different methyl marks on K4? Do you know how many, what fraction of the genome is methyl?
No, not yet. No, there's the citrullination field has been sort of, like I said, it had a flash, and then it sort of there's been discussions about its relationship to transcription. The absence of good antibodies has made life much more complex than it needs to be. PTM data in the early days wouldn't have seen it because it's the citronylation looks very similar in mass to some other modifications. But over the last few years, with the gains of mass spec, and there's been a lot of work on the citrullinome, and either in steady state cells or under netosis conditions, or after you flood the system with various recombinant enzymes. And there's a huge amount of work. And now that the mass spec has evolved, there's in fact there's even citronylation enrichment strategies. And now that the field has evolved in that way, what has been noticed, in fact, is that R8 and R17 came out of a mass spec data set from, I think it was a neuronal cell line about 6 to 12 months. I think the paper might just have appeared on bioRxiv. And so those I haven't read that paper too closely yet. I don't believe they have quantitative data in there. But now those marks are definitely present and in fact, we've seen it in multiple cell lines. And now what we're trying to figure out, and again, the biology is this real biology or is this cancer cell biology? Because remember what our citronylation does is it opens up chromatin structure. Cancer cells love open things because now that gives them access to switching genes on. Of course, they also like closing things down as well. But yeah, so maybe that's one of the things. Now we've only found out these antibodies work about 8 months ago. But yeah, it's a fun thing to carry on studying. The only thing is we probably won't do it because we're not planning on putting all of our R&D into citronylation.
Sure. So we got a couple of questions simultaneously about whether you've tested the lysine lactylation, or I guess along those lines, any of the other acylations. Do you know whether the specificity?
Yeah, we have not. When we made the acyl panel, we made acetyl butyryl and crotonyl at 927 and maybe 23, I think. We did not make lactylation. We did find, by the way, antibodies that are very good at crotonyl.
Yeah.
But we haven't screened the lactose. I would always be cautious as well because I have an antibody to lactylation at residue X, but inside the panel, there are no other lactyl forms. So the concern always is that what am I telling you? Am I telling you that it sees lactyl, or am I telling you that it's position-specific lactylation? Because now you have this false confidence. Yes, it distinguishes lactylation from unmodified or something, but that doesn't necessarily say that it's not swamped by something else. So we've always preferred to have panels of similar like modifications. Every program we've ever done, we've tried to represent as much biology as possible so that we're not the horrible phrase that's used internally is polishing a turd. that, you know, you're just making it look better. You're making yourself feel good, but all you've done is created data that doesn't really necessarily reflect what's really going on.
Right. So then I guess doesn't look like there's any more questions in the Q&A, so I'll just ask one more while we have a few minutes left. So you've this profile towards getting the entire PTM landscape of immune cells. Do you think this is something that other groups or consortia will be able to take forward, like in the Brain Initiative or in other parts of development? Or
I would say absolutely yes. I mean, so our plan for that data set was there's two manuscripts being prepared. There's one that talks about the quality of the data set. So that's the genomics, the automated genomics development. And that because it's in manuscript A, because it leaves space in manuscript B to say, okay, qualified data set, now here's the biology that can be done. There is nothing special about the immune system in this regard. I think all that happens in this consortium effort, and it was the ImGen guys with us and other support, is it sat back and it said at the very beginning, I'm not simply trying to create data ostensibly to a target. It sort of said, okay, I'm really, really trying to identify the antibodies that work and don't, the best ones. No antibody should sneak into the data set, no corners. So in other words, we could have generated canine tri data, but it wouldn't have been canine tri. And so we said, well, what's the point? These reagents exist, especially now that there's a lot more of them. It's eminently capable to find an antibody to a target. You just need more candidates at the front end. You need to hold a line at what your quality metrics are, and you need to be transparent about any limitations of the data in any consortium level effort. The limitations of the data, not simply that it is this thing, but that here's why I say it is this thing. But we've shown it can be done, and we're just one small company. But absolutely, consortium NIH level efforts could make the decision to do this. It's a lot cheaper to do it now than it was even years ago.
Yeah. Well, with that, we have hit the hour. So once again, thank you, Alan and Michael, for joining us. And I believe both of them have basically already agreed to have their videos published, so you can view it for a second time on YouTube in a couple of weeks. So thank you, everyone, for attending, and we'll see you again in 2 weeks' time.